Green works. Welcome to the Gold sponsor talks, and I just want to mention in the AIR community, we get a lot of support, a lot of financial support from our industrial sponsors, but also want to mention that a lot of the working group members and a lot of very active members of the AIR community have come from the industrial partners, and we continue in that tradition, and the working groups and subcommittees are all open to both academic and industrial partners. And we're trying to get some slides put up, but our first talk is from Selecta by Alex Chenchik, and as soon as we get the slides, we'll get started.
To switch your attention, and we are going to talk about technologies, not about science, so it will be more practical, how to use different technologies for immune repertoire profiling and immunophenotyping. And as introduction to the technologies, we just developed three types of technologies, and first is definitely bulk profiling, shown here, AIR, repertoire profiling assay, profiling of the in genome-wide scale, or for any focused set of the genes, like shown here in the middle. So it's so-called driver map assay, and profiling in the sorted T or B cell cells in the 96 well plates. All these technologies can be used for bulk profiling, for immune cell fraction analysis, or profiling in the individual for single cell analysis. As introduction to the technology, all these technologies based on the multiplex PCR. It's actually not multiplex PCR. It's based on the extension of the mixture of the forward primers, like reverse primers, like shown here, forward primers, and it can be applied not only for T cell or B cell receptors. You can really design primers against any human genes, and you can use DNA or RNA. And in this case, we just don't use multiplex PCR. We do extension from reverse primers, from forward primers, and after using universal amplification, using anchor primers from the both sides, and to amplify short region between, for example, 100 base pairs to maybe 600 base pairs for full length profiling of the immune receptor repertoire. In this example shown here for AIR assay, and what good in this assay, we design primers, validate them using several rounds, incorporate several mechanisms for primer dimer formation or non-specific amplification, and we can amplify all 7 chains in one reaction. For RNA, for DNA, usually it's required split TCR and BCR for 2, because cells are present in different amount, and we design primers only against functional isoforms. Both primers for DNA and RNA incorporate universal molecular indexes, which are very critical when you're dealing with so high complexity of molecules like immune receptors. And for data analysis, we integrated this with MyXCR software package. In order to facilitate really quality control analysis of all assays, which are developed currently for adaptive immune receptor AIR-seq assays, we design and make a panel of individual constructs, which can be RNA or DNA, which you can spike in any biological sample in order just to run multiple quality control analysis. And we developed 48 RNA spiking controls for BCR and 39 spiking controls for TCR, which are present the most commonly highly expressed isoforms of T cell and B cell receptors. And definitely this type of spiking controls, as I mentioned, they are universal. They practically cover full-length copies of transcripts for both TCR and BCR, and they can be very useful in order to not compensate, but actually measure amplification and sequencing biases in protocol. They also can be used in order to measure sensitivity and linearity in detection of different molecules. And for example, using these standards, we show that this type of assay, which I showed you before, RSA, has approximately detect 50% efficiency of detection of different mRNA for different TCR and BCR molecules. And what's good that all sequences which we design are already incorporated in MyXCR software package. So if you try to use it, you can really, it's very easy to use this type of the standards in order to see how assay what you developed work well in using these standards. If you try to use RSA and try to take whole blood or PBMC, you can generate data similar shown here, like here in this slide. From 50 nanogram of total RNA, you can easily identify approximately 100,000 different clonotypes for different chains, and number of the molecules what you detected in whole blood, it's rather balanced for different heavy and light chains, like shown here on this slide, which is rather useful because you don't need to split to amplify separately any chain. You can get information about all 7 chains in one experiment. The question is, can we get more information from this type of the simple experiment bulk profiling of immune receptors? And in order just to illustrate, we just run experiment. It's 2 immunizations, and you can see here it's typical immunization when you start to be older. And it's actually, I was the main person who really ran through this immunization. So we collect samples, multiple blood samples from me, and it was triple vaccination against pneumonia, tetanus, and diphtheria in first one, and another shingles. It's about shingle vaccine. And you can see here, it's like we run both genome-wide expression profiling, single-cell analysis, and immune repertoire profiling. And if you run genome-wide expression profiling, like shown here on this slide, you can see at approximately 2 days after first vaccination, you have very strong activation of innate immunity pathways, and approximately at 1 week, you can see strong activation of adaptive immunity. So it's rather obvious that when you just try to do adaptive immune receptor profile RSA using DNA and RNA samples demonstrated here on this slide, for the B-cell receptors, you can see very strong activation of, at the same time, approximately 1 week after vaccination, you can see strong activation at DNA level of multiple T-cell receptor sequences, clonotypes, and which correlate with the RNA level. So here we have, so it's indicated that we have both proliferation of the B cells and at the same time activation at the transcriptional level. For T cells, we don't have dramatic activation of proliferation of the T cells, but we have significant activation of T cell receptor approximately at the same time as B cell receptors. We speculate that this type of the data can be useful in order to identify activated clonotypes, not only for vaccination, but for many other biological experiments. And in this case, you just need to normalize RNA-based air profiling data to DNA-based profiling data. In this case, you can easily identify activated clonotypes. For B-cell receptors, it's easy. You have activation approximately up to 1,000, even sometimes 10,000-fold, which correlate probably with proliferation of plasma cells and strong production of antibodies and B cells. For T-cell receptors, it's also correct, but activation level is significantly less. It's between 50 to 100, up to 50-100-fold activation, but still it's enough in order to identify cells which are activated by immunization or any biological process which induced adaptive immune responses. It probably doesn't work in all cases, but at least in this case, in many cases, this strategy, simple strategy, is really rather useful in order to identify activated clonotypes. We also even try to use immune watch detect algorithm, which you hear presentation yesterday, and actually we were successful to identify epitopes which recognize by vaccination to Shingrix vaccine. And in this case, you see we identify very clear that glycoprotein E from zoster virus, it's one of the main targets for this vaccine. Based on the sequences of T-cell receptors. Basically, this identification of activated T-cell receptors, it's only first step in the journey of trying to understand specificity of these receptors and try to correlate this type of the cells which express these receptors. The main strategy really to link phenotype of the T cells or B cells with specific receptor sequences is just to use 10x genomics, which is the most popular technology shown here on the top. And what is interesting in this technology, you just sort the cells in droplets, generate single cell expression profiles, cluster them together using t-SNP plot, and identify clusters of these connect these clusters of the cells to specific cell type using cell-specific markers. From this data, you can generate both expression profiles for each cell and expression of the T cell or B cell receptors. Interesting, the same type of the data can be generated rather simple because you can use the same markers which you use to identify cluster of the cells, just sort the cells in the test tubes, and from these test tubes you can generate both air profiling data and expression profiling, for example, for 500, the most popular T or B cell markers. In this case, you can generate very similar information, but doesn't require any really expensive instrumentation. So if you have small number of the cells, and it's very typical, for example, if you try to sort activated cells or cells specifically bind to some specific peptide MHC tetramer, in this case, you can sort the cells in 96-well plate and run assay what I described here, which is based on the expression of the T cell, B cell receptors and 30 markers. Useful, this technology is rather simple, and what is good here, you can sort the cells in 96 plates, store in -80 refrigerator for any period of time, and run this assay without any special equipment. And last slide is acknowledgement. It's a summary of what we discussed today. We discussed today development of the bulk air assay for bulk profiling. And we discussed possibility to detection of antigen activated clonotypes based on correlative analysis RNA and using DNA and RNA data for the same cell types. We discuss strategies for immunophenotyping of the cells based on the profiling in immune cell fractions and in sorted T cells. And we discussed development of TCR and BCR control constructs. And if you're interested to use this technology, please contact us. All these technologies, what I described, are available as a kit or custom services. Thank you very much for your attention.
We'll have time for questions, but
Yes.
of course, all our sponsors have
Yes, we
exhibits
have-
in the hall.
Yes, we have booths. If you have any questions, please ask.
It's green. I think it should be on. Oh, I'm not speaking into it. And so the next speaker will be Victor Greif from Imprint, an exciting new initiative that he is one of the co-founders of.
Yes, thanks for having us in this session, and it's a great pleasure to finally introduce to you all Imprint, which is a focused research organization for immunology, of which I'm one of the co-founders, and I've taken on the director role in computational immunology. So what is Imprint? Imprint is a focused research organization called also short FRO, and we are focused on decoding the language of the immune system to identify hidden causes of disease. I will brief, so we will be launching officially early fall 2024. You are the first to know about us, but we are currently still in stealth, so we would ask you to please don't share any of those slides on social media. So what are FROs? FROs can be thought of non-profit startups. They have an impact-driven mission. They develop field-enabling public goods. They are enabled by philanthropic funding. They operate at the intersection of academia and industry. And they have a finite timeline of about 5 years. And what is, I think, really important for us to collaborate with this community is that we have non-profit startup status, which facilitates the collaboration with academia. How are we supported? We are supported by Convergent Research, short CR, and Convergent Research is a non-profit itself, which has as its main mission to developing, promoting, and actualizing focused research organization structures. And CR is a member of the Schmidt Futures Network and has funded to date 8 FROs. So where does this inspiration for this entity of FROs come from? It comes from this idea of large-scale coordinated research programs in the support of big engineering in support of science. So you can think of it as super large-scale projects such as the Hubble Space Telescope, but also the DARPA Grand Challenges, for example. And it really focuses on developing tools that are then disseminated to the public and which should then enable novel discoveries. So to make this a bit more specific, FROs exist really at the intersection of the more conventional entities such as corporations, academic research, and startups by focusing on, and this is shown in bold here, large-scale efforts to create public goods, but in tightly coordinated focus teams. And the nice thing, for example, as opposed to academic research, is that we are not tied to funding cycles or other incentives in academia that are really a barrier to coordinated engineering teamwork. And as opposed to industry, which is mostly not incentivized to work on non-commercializable projects or shared resources. So our tools that we will develop will be shared with the community. So what is our scientific question, very broadly? So you all know that that knowledge about disease is required to design therapeutics and to treat it. And though we might not know what causes a chronic disease, our immune system probably does, as we all believe, with AIR sequencing. And we all know that immune memory stores a distributed memory of immune exposures. And what we are trying to build is tools to read out these memory signatures and to reconstruct timelines of disease to identify ultimately hidden causes of disease. So you can think of this as there are many examples for this here. I show 3, but there are very famous trigger-target relationships of what we, which we would like to, for which we would like to develop tools to link trigger to target on the immune memory level. So we want to use forensic immunology tools to reconstruct immune histories, to reveal hidden etiologies by focusing mainly on the immune repertoire. So as an example approach, we would then combine experimental tools such as high-throughput sequencing, antigen binding assays on the experimental side, cellular timestamping, and then develop novel computational tools, as I said before, to reconstruct those exposure timelines. And then once we have those tools, to apply them to disease models at a very large and robust scale. Reconstructing immune histories, we are mainly interested in identification of novel causes of disease, but of course this can be used for diagnosis, prediction of disease outcome, or ultimately, of course, where many of us want to go, modification of disease outcomes. To summarize, we are a non-profit building experimental and computational AIR-seq research tools. We have the shared objectives with the AIR community to disseminate our validated AIR research tools to the public. There are many potential areas of interaction, collaboration on tool development and data set generation. We are looking for technical advisors and advisory board positions. We are looking also to hire from within the AIR community, and we are looking also to contributing to the AIRC working groups. So the pictures that are not grayed out, those people are actually here at the conference, and probably you have met already many of them, my co-founders, Beck and Raymond. and also the computational team, Tim, Julien, and Julio are all here, so we'll be happy to talk to you. So yeah, please come talk to us. We have a very bare bones website where we have a contact scheme, and also some job announcements are on there. So, and thank you very much for your attention.
Well, great. We're actually back on time, so unless any questions from the floor, but why don't we just get going? So next we're going to hear from My Labs, and we have Stan Pozlowski, the CEO, who's going to tell us about My Labs. Thanks.
Yeah, but I need to connect my laptop. To share. Okay. Good. Let's see. 1 second. That's no, that's not. So, can you just 1 second, let one more? And then just 1 second. Yeah. So, second. Yeah, so it works. So, thank you very much. My name is Stan, and I'm Chief Executive of My Labs. Many years ago, I was one of the initial creators of our MyXR software, so I wrote a lot of lines of code. But today we have more than 20 software engineers and bioinformaticians constantly working on the development of our software platform. And most of them actually came from academia with many, publications in the science. And tomorrow our Chief Science Officer, Professor Chudakov, will give a talk about, scientific things which we are still related to. We are very proud that MyXR is quite popular today. We are proud to provide it absolutely free for all academic community, and at the same time, we work a lot with pharma companies, so many of the big pharma companies use MyXR. And just one number, during the past 7 years, more than 10 million of samples were processed with MyXR, which is in fact a huge number. So today we have more than 1,000 of organizations using MyXR constantly for the immune research, and we work we have our very good users in many of the universities and research organizations. And as I mentioned, we work with biopharma companies who develop the breakthrough therapies today and use MyXR, and we are, of course, very proud to contribute to these things happening today. So, in this talk, I wanted to briefly highlight what are the new things in MyXR, because the first version of MyXR was released in 2015, 9 years ago, and many people still use the versions from 2017, 18, etc. So the main purpose of my talk today is just to briefly highlight what are the new things in the recent versions of MyXR. And the first thing, and that's, the key thing from the first versions of MyXR, that it is very flexible, and it works with any kind of sequencing data. So whatever type of the data you have, you can use MyXR. In the recent versions, we made a specific attention to support, like a specific pipelines for all of the modern kits and sequencing technologies. So in the recent versions of MyXR, we have a dedicated and simplified support for all of the popular reagent kits used by the scientists, like Illumina kits, Takara kits, Selecta kits, and our own kits as well. And we support all types of the analysis, like with DNA, RNA, with or without molecular barcodes, single cell, etc. So I'll cover this briefly in the next slide. And at the same time, as usual, if you have any custom library preparation protocol, then you can adapt MyXR pipeline to use it with your sequencing protocol. So the first thing I wanted to highlight as a new part of MyXCR platform is that now we have two parts of the pipeline. Initially, when we released MyXCR, it was focused on the upstream analysis, where you have your FASTQ files, you run MyXCR, and as a result, you have tables of the clonotypes. And then you can use, and many people used other tools like VDJ tools, also developed in our lab for the downstream analysis. And now we support all downstream analysis capabilities right in the MyXCR. So you have two parts of the pipeline. The first part, when you start from your FASTQ files, and then also a new thing that we support barcodes, any kind of barcoding, UMI, cell barcodes, sample barcodes, etc. And as a result of the upstream analysis, you have your tables of clonotypes. And then the second part of the pipeline is the downstream analysis, where we have all of the standard features for the basic downstream analysis, like downsampling different, let's say, repertoire profiles like gene usage, diversity, overlap, and also you can even use MyXCR for visualizations and will generate plots, like, for example, for time trackings and other things. The next big thing, which we released 2 years ago, is single cell. Well, obviously, more and more people are using single cell. And in MyXCR, we have a dedicated support for all of the modern single cell technologies, like 10x Genomics, BD Rhapsody, and ParsBio, and whatever, including like low throughput technologies like plate-based, etc. And in fact, well, it took us like a year of the development of many people and efforts to really implement a very good and reliable pipeline for the analysis of high throughput single cell data like 10x Genomics, because there are plenty of peculiarities and technical things under the hood, like, for example, how to filter cells, how to, well, find the real cells, etc., etc. And we are proud that right now our pipeline works like, in many cases, it even works better than Cell Ranger for VDJ data, especially in the case where the quality of the data is not great. So the last thing I wanted to cover as a huge addition to MyXCR is that recently, well, last year we released a dedicated support for the antibody, well, different pipelines and different ways how people are doing antibody discovery. And in the recent versions of MyXCR, we have a specific functionality to identify the antibody candidate leads, both on the clonotype level of the homology clusters, and somatic hypermutation trees. And in fact, B-cell lineage reconstruction and, well, somatic hypermutation trees is one of the very important parts of modern MyXCR. So those are the three things I wanted to highlight about the MyXCR and the recent developments of MyXCR. But in general, what we see, so we have more than 2,500 laboratories using our software, and most people who use MyXCR and who actually do repertoire sequencing are actually biologists. People who are not handy with a command line and things like that. And MyXCR, as you know, is a command line platform. And here we have a lot of users who are bioinformaticians and very happy with the command line version. But what we see that people also need a graphical user interface to do the interactive discovery, data discovery process, to find the insights and to find the answers to the R&D questions that they have in the data. And because of that, we recently, just a few weeks ago, we released the first version of our new platform. It's called just Platforma. And the key thing is that it provides, as a part of the platform, it provides an interactive graphical user interface. And all things that you were able to do with recent versions of MyXCR and the command line are now available from the interactive graphical user interface, which is designed specifically for biology people. And the interface speaks in the biological language. It's very important to understand that, well, we understood this, and we see in our users a lot that repertoire sequencing is not, well, the one thing which people do even in this field of repertoire sequencing, there are plenty of applications, plenty of algorithms, and many things. So that's why we are building not a, let's say, a graphical user interface around the MyXCR, but we are building an open platform which supports all sorts of applications in the computational biology, starting, of course, with the repertoire sequencing. And the fact that the platform is open allows other people, let's say bioinformaticians and bioinformatic developers from academia, for example, to develop their pipelines, their blocks, and bring them to the platform to be able to allow scientists and biology people use those pipelines and algorithms from the nice graphical user interface without like the need of running the code. So, and as well as MyXCR, we provide all our software absolutely free for academic research. So that's it. Just one thing. We have our bioinformatics support team, people who are working just to help scientists to do their research. And you can reach our support team at support@mylaboratories.com with whatever questions you have related to MyXCR platform or anything else. Thank you.
Great, very exciting. Oh, maybe we can do one at the most or two. How do you do time tracking? What is the core intuition for how Mixer compares between time points?
So, there are two parts of the time tracking. The first part is to identify the clonotypes which you want to track. That's the first part. And the second part is just the time tracking is just a visualization where we put the abundances of the clonotypes on the plot. And the first part is a tricky. The second part is simple. To identify the clonotypes which are, let's say, interesting to track, we have like algorithms for, we call them differential clonotype abundance to, well, find the clonotypes which show, let's say, expansion in their abundance between different metadata points like time points, etc. So I can explain a little bit more detail at the booth, which we have here.
Yeah, I think we should move on because we've got two more.
Yeah.
But, our next is from Pipe Bio with Yannick Benson, and a longtime supporter of the AIR community. Thank you.
We'll try. Well, first of all, it's really great to be here. It's great to hear all the talks around data standards for the AIR community around, yeah, as much as possible. To hear about, machine learning, how you organize your data, so it can be useful for machine learning. And this is basically what we have tried to encompass in one big platform. I was promised 7 minutes, but we're already over time, so I'll be extremely fast. Please come to our booth for additional chats. We'll be more than happy to take you through this in more detail. Most importantly of this talk will be to mention that we'll do an antibody athletes run tomorrow morning, 6:30. So if you're up for running, then please come talk with us, and I'll give you instructions where to meet up. So anyway, so what is Pipe Bio? It's an integrated bioinformatics platform for scientists. And no offense, but when we talk about scientists, we mean the wet lab scientist doing a lot of the data generation, and we're very much sequence focused, and this is what I'll basically talk you through. So we have this bioinformatics platform. We can basically annotate everything, so we have heard a lot about full immunoglobulins here and TCRs, but we have also support for single-chain FVs, VHHs, and also other quirky modalities such as Affibodies, Knottins, and a lot more. So in the platform, I'll show a lot of screenshots from the platform, but you can do a lot of QC on your data and analysis, so annotations, enrichment, hit picking, and so on. So where do we sit sort of in the ecosystem? So we sit sort of in the middle where from the wet lab you generate a lot of sequence data, that being NGS sequence data or Sanger sequence data. We also have support for 10x, PacBio, Nanopore, and a fairly sequence agnostic. That data is brought into Pipe Bio either via the API or by drag and drop. Wet lab scientists do not like to use the API, so they typically use drag and drop. We do all the analysis, they analyze a lot of the data, they do all the hit picking. When they have done that, they can register their sequences in BenchLink, LabKey, GeneData, whatever they prefer from an ELN perspective. Then they want to do additional characterizations, probably with Cartera doing some SPR and whatnot. And there's basically a feedback loop back into PipeBio or some of the other registration systems. So just about the format, so I just mentioned before that we have support for all of these modalities, TCRs, bispecifics, immunoglobulins, peptides, and whatnot. On top of that, as part of the platform, we also have germlines for all the species, so it's very easy and quick to get up and running. It should only take a few minutes, basically. On top of the sequence data, we also have support for functional data, so you can also add your ELISA data, your SPR data, or whatever other types of data you actually have, and you can associate that with your sequences. So these are all screenshots from the app. As part of the core annotation pipeline, we of course identified the closest germline gene. We look for somatic height mutations, and we also look for developability issues. So there's a lot of PTMs to be detected as part of the run. All of this can be scored individually, so you have very easy ways of filtering down the data to actually hit pick your final candidates for a good antibody drug. We do in-app workflows, so you don't need to run command lines to actually string analysis together. So you can basically run this as part of a workflow which can be configured. This is very convenient if you do repetitive tasks. So here as an example, we do QC, on the data we merge them, we annotate, we cluster, we can add functional data, and then basically you hit pick at the end, and then you can add them to your ELN or LIMS system. So all within the app for repetitive tasks, so very easy to use. Just a bit of eye candy here at the end. So the platform is very graphical and very easy to use. For all our charts and graphs, they are interactive, so you can use the graphs to filter on the data. So as an example here, we have an amino acid bar chart. So if in one position of your CDR, you want to look for a particular amino acid, you basically click that chart, and that'll list down the data. As part of the platform, we of course also have clustering and clonotyping as any other sequence analysis platform in this field. We also do have a database component of the platform. So if you do several campaigns, you can deposit your hit-picked sequences in a database, and then you can basically ask the sequence, have I ever seen this sequence before? So very powerful and valuable. Cluster networks, of course, it looks good. It's also very valuable, so when you overlay functional data to the clusters, you may also use this for hit picking. You don't want to use all the sequences from one cluster if you can only do further characterizations on a single one. Alignments, very powerful. Here we just have an alignment with some functional data attached, also showing hydrophobicity, so you can also look for hydrophobic patches. Phylogenetic trees as part of the alignment. Again, here we have a circular representation where we have overlaid a lot of EC50 scores, also for easier hit picking and visual interpretation of the data and the sequences associated. And here the same data, just in a sort of a linear view. Here we have a pairwise distance matrix, so you can also see how different are the sequences that you are finally hit picking. And of course, we have here a circus plot, which is looking very close to our PipeBio logo. And not surprisingly, heat maps, if you have several rounds of or several samples, you can also compare this in a pairwise or in a heat map manner. We use A-Body-Builder 2 from Charlotte's group to do structure prediction, but that's also part of the app. And of course, for the bioinformaticians here, we do have a REST API, so you can actually execute code both on our platform, but you can also use the API to import and export data. There is a GitHub repo, but you will not have access until you have an account on the platform. Thank you.
I think we'll move on because we have one more, but thank you, Yannick. And now we have Takara Bio, and we have Shavita Goyal, and thank you for sharing with us.
You can hear me, correct? Okay, so thank you everyone and a very good afternoon. I think you all are ready for a break now, but just hear me for a bit. I will be super fast. So I'm Shweta Goyal, I'm from Takara Bio. Before I start my talk, I just want to give a brief introduction about Takara Bio. So I think a lot of you already know about Takara products and are using NGS products, but we are not just limited to NGS. We provide whole range of cell and molecular biology reagents ranging from PCR, cloning, nucleic acid purification, gene delivery, functional genomics, and because of our world-class products, we also OEM these products to molecular diagnostic labs. So focus of my talk today is about immune repertoire profiling, more specifically BCR. And in immune repertoire profiling, we provide solutions for both B cell receptor repertoire and T cell receptor repertoire, which are compatible with both human and mouse RNA samples. Some of the kits are also compatible with rhesus RNA samples. Our kits has a flexible input range, which range from 1 nanogram RNA to 1 microgram RNA, and they're compatible with various sample types, be it B cell or T cell RNAs, PBMCs or whole blood or primary lymphoid tissues. We have UMIs for error correction, which help in the confident clonotype calling. The sensitivity of kit is pretty good, and we can capture low abundant clonotype. Some of the data I will be showing in the later slides. For BCR, we identify all the 3 chains, heavy, kappa, and lambda. And for TCR, it's more focused on alpha and beta chains. We provide sequencing flexibility also, so user can do CDR3 only sequencing or full length VDJ CDR3 based on the question they want to answer. And for the multiplexing, we provide UDIs, which can help multiplex 384 samples in one single run. Now, we have been hearing about diversity of immune repertoire, and it's important, and it requires accurate analysis for correct biological information. And there could be biases from assays like PCR or multiplex assays or sequencing platform biases. So this study is done in collaboration with FDA. We like to thank Wan-Ming Xiao for including us in this collaborative effort. So the aim behind this study was to understand the gaps in the BCR immune repertoire profiling and to provide the controls to make the analysis more understandable. So we received the RNA samples from FDA, which are PBMC RNA and cell line mixed RNA. So 9 cell lines were mixed in the different proportions, ranging from 35% to 1%. These cell lines are spiked into PBMC at 5 different dilutions. And the lowest concentration of the clonotype in the D5 dilution is 1 in 1 billion. So we prepared the libraries using our SmartSeq Human BCR kit, and I think most of you know about the workflow. It's DT primed RT step, and in the RT step, with the help of template switch oligos, UMIs are incorporated, which helps in the error correction in the downstream analysis. And followed by that, there are 2 semi-nested PCRs, which result in the final library. The sequencing was performed on Illumina platforms, NextSeq 2000, MySeq, NextSeq 2K, and also some of the libraries were sequenced on Element AVITI system. The data was analyzed using Cogent NGS immune profiling pipeline from Takara Bio. Apart from doing the NGS assay, we also did the ddPCR assay. So for the ddPCR assay, the assay was designed using the sequencing shared by FDA for the cell lines. I'm here only showing the heavy chain data. From the ddPCR assay, we saw that JK6 is the most abundantly expressed cell line, followed by WSU, DL, CLC, which was later, this data is also correlated or also is similar to the whole transcriptome and whole genome analysis done by FDA along with Illumina. So before I start discussing the spike-in data, I want to show here that we prepared the libraries with 25 nanogram, 100 nanogram, and 500 nanogram RNA inputs in triplicates, and we see an increase in clonotype counts as we increase the input, and we identify all the 7 isotypes for the BCR. And there is a good correlation between the technical replicates as the clonotype frequencies correlate between the low and high inputs. So this graph on the left shows the abundance of cell lines from NGS data. We see that JK6 is the most abundant cell line from the NGS data, while JK6 is the second most highly spiked in. It's spiked in at 25%, while MAC1 is spiked in at 35%. But it makes sense because JK6 is found to be expressing higher as compared to other cell lines, and if we normalize the ddPCR expression data with the proportion of cell lines spiked in, we see that the proportion of observed cell lines is comparable to the expected cell lines. So that means there is no bias as introduced by the assay, by the PCR primers, or by the sequencing for which can affect the proportion of the cell lines as observed in the NGS assay. Here I'm showing the linearity of dilution. So 9 cell lines were detected in the NGS assay, and there is good linearity of detection for all 9 cell lines, which means that the number of UMI groups identified at tested concentration is dependent on the copies of the target molecules. So again, which supports that there are no biases introduced, and there is good correlation for all 3 inputs tested with more than R-square with more than 0.9. These graphs are showing the sensitivity, and we see that at 500 nanogram, the limit of detection is 4 times 10 to the power minus 7, and this is 500 nanogram input and 15 million reads. And I believe if we increase the input or increase the sequencing depth, we can reach to 1 in 1 billion limit of detection. Illumina also did only 25 nanogram libraries comparison on MiSeq, NextSeq, and NextSeq XLeap, and we the sensitivity across all 3 platforms is very much similar, which shows that the assay is agnostic to the sequencing platforms. As I said in the beginning, that we also did some libraries and sequenced on Element Biosystem IVT. And I'm showing here the data for 500 nanogram, and it also shows good linearity across all 9 cell lines, and with the correlation coefficient of more than 0.95. And again, it shows that the UMI groups identified corresponds to the target molecules in the sample. And this is the sensitivity data from IVT system. And here also we see it's comparable with Illumina platforms. And again, the limit of detection is around 4 times 10 to the power minus 7 at 500 nanogram RNA input. With this, I would like to conclude, and I would say that the assay is free of biases because we see that there is correlation between the ddPCR expression data and NGS data, and we see that there is reproducibility between the technical replicates. there is accuracy in detection of the clonotypes because we see that the abundant clonotype should be JK6, and we see that same in the NGS data. There is good sensitivity with limit of detection less than 10 to the power minus 6, or we can increase it to even further by increasing the input. And the assay is agnostic to the sequencing platforms, so we do not see any differences or any change in the sensitivity or accuracy as we move from one instrument to another. With this, I would say that's good science, and if you have any questions, I will be happy to answer those.
It's nice to end the session on such good news and positive results. I think we need to wrap up because we're having a new session in here at 30 after the hour. But let's thank the gold sponsors and all of our sponsors for our